Hello everyone. In our previous video we have studied about the megasporangium and different types of ovules. In this video let's study about the development and the formation of the female gametophyte, that is the embryo sac. Let's recall the structure of the ovule. So we have the stalk and the body. The stalk is called as the funicle and the funicle extends over the body with the help of the raphe and the stalk is connected to the body with the help of hilum. Inside that, this segment, with these segments are called as the integuments. And the integuments arise from chalaza and below there is an opening which is called as the micropyle. Inside the integument there is a dense mass of diploid tissue and this diploid tissues are called as the nucellus. Now let us consider this particular cell and this is called as the megaspore mother cell which is towards the micropyle region. Now, this megaspore mother cell is diploid in nature. This is called as megaspore mother cell. We use the same MMC in order to represent the microspore mother cell or the pollen mother cell. This megaspore mother cell now undergo meiotic division. It undergo meiotic division to give rise to 4 haploid megaspores. It gives rise to 4 haploid megaspores. All the 4 are haploid and they are called as megaspores. Now, this is called as the linear tetrad. Whereas, in the previous cases, that is in the microspore formation, we have seen the tetrahedral form. Whereas in this we have the linear tetrad of haploid megaspores. This is the chalazal region, this is the micropyle region. Similarly, this is the chalazal region, this is the micropyle region. It is very important to label the chalazal as well as the micropyle region. The 3 cells, the 3 haploid cells towards the chalaza degenerate. The only cell towards the micropyle survive. And this is called as the survival megaspore. Now, this survival megaspore, which is haploid in nature, undergo meiotic division. It undergoes meiotic division. To give rise to, first in meiotic division, karyokinesis takes place followed by cytokinesis. Here, 3 karyokinesis take place. 3 successive karyokinetic event takes place. In the first karyokinetic event, there is a formation of 2 nucleus. One in the chalazal region, the other in the micropyle region. In the second karyokinetic event, there is a formation of 4 nucleus. This is the previously formed nucleus. Now the second set of nucleus gets formed. This is the second set of nucleus. In the third karyokinetic event, there is a formation of, so this is the previously present ones. This is the previously present nucleus. Now in the third karyokinetic event, there is a formation of another 2 nucleus. So this is the karyokinesis, 3 karyokinesis event which is taking place in the survival megaspore. The first karyokinesis, it is 2 nucleated. In the second karyokinesis, it is 4 nucleated. In the third karyokinesis, it is 8 nucleated. Now, one from the chalazal region, one from the micropyle region migrate towards the center. Now, we can see the cell here. One from the chalazal region, one from the micropyle region moves towards the center. Now, this nucleus is called as the polar nuclei. Now, the remaining 3 cells in the chalazal region, and remaining 3 cells in the micropyle region undergo cytokinesis. Now here the cytokinesis event takes place. Now we have 3 cells here and 3 cells here. Now this develops cytoplasm around it and it forms cells. The 3 cells towards the chalazal region and 3 cells towards the micropyle region. These 3 cells are called as the antipodals, and the 3 cells towards the micropyle region are together called as the egg apparatus. The small cells, these 2 small cells are called as the synergids. And the bigger one is called as the egg. So let's recall this. In the nucellus we have 1 cell towards the micropyle, which is called as the megaspore mother cell. The megaspore mother cell is diploid in nature, which consists of the chalazal region as well as the micropyle region. Now this megaspore mother cell undergo meiotic division to give rise to the linear tetrad megaspore, where all the 4 cells are haploid in nature. The 3 cells towards the chalazal region degenerate and the 1 towards the micropyle region survive, and this is called as survival megaspore, which is haploid in nature. This survival megaspore undergo mitotic division, which involves a karyokinesis and cytokinesis, whereas 3 successive karyokinesis is being followed by the cytokinesis. In the first karyokinetic event, it is 2 nucleated. In the second karyokinetic event, it is 4 nucleated. In the third karyokinetic event, it is 8 nucleated. So, we have 4 towards the chalazal region and 4 nucleus towards the micropyle region. 1 from the chalazal region, 1 from the micropyle region moves towards the center. Now, these 2 nucleus are called as the polar nuclei. These polar nuclei fuse to form, it fuses to form the secondary nuclei. Now we have 3 nuclei at the chalazal region and 3 nuclei towards the micropyle region. Now they form cytoplasm around them to form a cell. Now these 3 cells towards the chalazal region are called as the antipodals, and these 3 cells towards the micropyle are together called as the egg apparatus. The 2 smaller cells are called as synergids and the large one is called as the egg. Now, this is called as the embryo sac. Now, this is called as the embryo sac. The embryo sac is developed from the single megaspore mother cell, and it gives rise to linear tetrad. And this linear tetrad, the 3 degenerate and 1 has been survived, and the complete embryo sac has been developed from 1 haploid survival megaspore. Hence it is called as monosporic. Hence it is called as monosporic. Now we will count the nucleus present: 1, 2, 3, 4, 5, 6, 7, 8. It is 8 nucleated. Let's count the cells: 1, 2, 3, 4, 5, 6. That is 3 antipodals, 3 egg apparatus, 1 complete embryo sac. That is, it is 7-celled. 3 antipodals, 1, 2, 3. Egg apparatus, we have 3, 1, 2, 3. 1 complete embryo sacs makes it 7-celled. Since it consists of antipodals, polar nuclei, egg apparatus, this is called as the polygonal. Thus, we say that embryo sac is monosporic, 8 nucleated, 7-celled polygonal type. It is monosporic since it is made up of a single haploid survival megaspore. It is 8 nucleated because it consists of 8 nucleus, 3 in the antipodals, 2 polar nuclei, 3 in the egg apparatus, it is 8 nucleated. It is 7 celled because it is having 3 cells in the antipodals and 3 in the egg apparatus. And this complete embryo sac consists of the antipodal, polar nuclei, egg apparatus. So we call it as the polygonal. So embryo sac is nothing else the monosporic 8 nucleated 7 cell polygonal type of embryo sac. Till now we have studied the formation and the development of the male gamete and the female gamete. In the next video we will study how these male gamete and female gametes are transferred for fertilization. Until then, happy learning from H2all.